Hello, students. Very good evening. Today, we will complete the rest of the topics that we have for the respiration in higher plants. So, we were left with two topics. In the last class, we completed with the Krebs cycle. So, today we will start with what happens after the Krebs cycle, correct? So, let's get started. If you have any technical issues, please let me know in the chat box so that we can correct it during the course. Okay. Yeah. So what happened oh sorry, what happened when we talked about, one second, yeah, hold on, I'll just post it here. Yeah. While we talked about Krebs cycle, some of the key points that we have to remember, as I told you before, one is the decarboxylation. What is decarboxylation? We know that during the Krebs cycle, what happens? We cannot directly use the pyruvate. So the body has a mechanism where it converts pyruvate, correct, right? So the pyruvate under the enzyme pyruvate dehydrogenase, by reducing NAD plus to NADH, we converted pyruvate into acetyl CoA, which is a starting material for our Krebs cycle. And in the Krebs cycle, what happened? The acetyl CoA, okay, which is in the matrix of the mitochondria, which is transported to the matrix of the mitochondria. Here, acetyl CoA combines with a 5-carbon atom oxaloacetate. Okay, so here a condensation reaction happens, and we know that we get citrate, and the citrate is isomerized to isocitrate. And the conversion of isocitrate into alpha ketoglutarate requires reduction of NAD plus to NADH. Very, very, very important. This is the first key compound. And during this, a decarboxylation reaction happens where a carbon dioxide molecule gets removed. And then coming to alpha ketoglutarate, is converted into succinyl CoA. So here also one more NAD plus is reduced to NADH. Again, one more decarboxylation reaction happens where another carbon dioxide molecule happens. So there are two decarboxylation reactions happening. Now coming to succinyl CoA to conversion to succinate. Here, look here, there is a phosphorylation reaction happening where the GDP is converted to GTP. So that is in animals, and then ATP is converted into ATP is converted into ADP down the further reaction. So this is one phosphorylation reaction. Okay, and then again succinate to fumarate. While we want to regenerate the entire process of converting the oxaloacetic acid, when succinate is converted into fumarate, FAD is reduced to FADH2. So the NADH and the FADH2 becomes a very two key ingredients for our electron transport chain. Okay. Now look here, fumarate was converted into malate, and the malate conversion to oxaloacetate again reduces one more molecule of NAD plus to NADH. So 1, 2, 3, three molecules of NADH, and then one molecule of FADH, and then you have one phosphorylation reaction that is happening where the GDP is converted to GTP. So this is what we understood with respect to Krebs cycle. Now, the outcomes of the Krebs cycle. What are the outcomes of this Krebs cycle? As I have told you, the energy that is stored in the form of NADH and FADH2 is used for a production of energy. So what happens here? If you look at the structure of our mitochondria, the mitochondria as the outer membrane. So this is the outer membrane of the mitochondria, and this is the cytoplasm of the mitochondria, and this is the inner membrane of the mitochondria. See, outer membrane, inner membrane. Between the outer and the inner membrane, there is a space which you call it as perimitochondrial space, or we also call it as intermembrane space. Okay, perimitochondrial space or intermembrane space. The difference between these two membranes, see, what is membrane? We have an organelle which is our mitochondria. So it has two layers. Okay, one is the outer layer which is continuous and permeable. So when I tell permissible, it allows many of the substances to cross through its border, such as hormones, enzymes, proteins, etc. Okay. Whereas the inner membrane, which is discontinuous means it's not like a circle. It is inner folded like this into lot of finger-like projections called as cristae, which divides our matrix into small compartments. Reason being, it increases the surface area for your reaction. Okay. What is happening in this inner membrane? Inner membrane has a job. What does it do? It makes sure that it has some of the elements that are embedded within the membrane, which we call it as complexes. So each of these complexes, so let's say we have 5 complexes. So in these 5 complexes, what happens is they are specialized to act as electron transport chain. So electron transport chain means they are carried from one complex to the another complex to the another complex. So during this process of moving these electrons, what do our inner membrane do? Inner membrane starts accumulating. Okay, it starts. See here, you can see here hydrogen, hydrogen, hydrogen. So it starts accumulating a lot of protons within the intermembrane space. So this proton accumulation is what we call it as the proton gradient. So when the proton gradient is created, now we know how the ATP synthesis will happen. So let's see how this series of electron carriers, which are like 7 electron carriers, are present within the inner membrane, and what are the conditions and how they function. Okay. See, when we look at the 7 important electron carriers that are present in our inner membrane, first one is NAD, that is nicotinamide adenine dinucleotide, NAD, which is present in the complex 1. Next one is FMN, that is flavin mononucleotide, okay, that is present in complex 2. Then we have flavin adenine dinucleotide, that is also present in complex 2. Then coenzyme Q, which is also called as ubiquinone, which is a carrier. Then we have cytochrome B and cytochrome C. So cytochrome C is something called as a mobile element. We'll talk about it. And then we have cytochrome A and cytochrome A3, which is the last elements of our complex 4. Okay. So these are the names of our complexes. So remember, students, if the question is asked about what is present in complex 1, you should go for NADH, CoQ, and then we have oxidoreductase, right? If they ask about complex 2, then we said succinate dehydrogenase. Remember these names, okay? What do complex 2 have? They have succinate dehydrogenase. What do we have in complex 3? In complex 3, we have cytochrome BC1 complex. And what do we have in complex 4? We have cytochrome C oxidase. And then what do we have as the carriers? One, we have CoQ, that is coenzyme Q, or which is also called as ubiquinone, very important. And other one is called as cytochrome C, very, very, very important. So, okay, I will tell you about what to remember in this, so it becomes easy. So what is happening here? So here, something very important happens. So first thing is we have the complex 1. In the complex 1, what happens is NADH is oxidized to NAD+. When NADH is oxidized to NAD+, then it releases 2 hydrogen. Okay. During this process, it donates 2 electrons to our complex 1. So now what complex 1 do is, once this bond is broken between NAD and H, so now the electrons are taken up by the complex 1 and it is given to our ubiquinone, which is called as CoQ. Okay, it receives the electron for CoQ. So at the same time, what happens as we oxidize this, now we have the movement of hydrogen atoms. To move this electron, see, it was in complex 1, and to give this electron to ubiquinone, now what happens is 4 of the protons or 4 of the hydrogens are pumped into the inner intermembrane space. Okay, that is the first thing that is happening. That is complex 1. Now, if you come to complex 2, in complex 2, what is happening? In complex 2, FADH2 is reduced to FAD. Here also 2 hydrogen atoms are released. So, if you look at the complex 2 here, it has something called as succinate dehydrogenase. Succinate dehydrogenase, what it will do? It will convert succinyl CoA to succinate. So, as during this conversion, if you look at the Krebs cycle, you will understand. So, it comes during the Krebs cycle. What is happening here? We have succinyl CoA, okay, and then it is converting it to succinate. Or you see here, it is converting into fumarate, succinate to fumarate. During this process, you can see that FAD plus FAD is reduced to FADH2. The same enzyme, okay, succinyl dehydrogenase. Hold on 1 second, students. Okay, the same dehydrogenase, what it does is, okay, you can see here, it will oxidize FADH2 to FAD. So once it does that, it donates again 2 electrons, and the 2 electrons, to move these 2 electrons, what happens? It gives the electrons to the ubiquinol. So ubiquinone, okay, ubiquinone is converted to ubiquinol. Now, what ubiquinol does, it took the electrons from complex 1, it took the electrons from complex 2. Now it donates this complex 1 and complex 2 electrons to complex 3. Now, once it goes through the complex 3, we know that our complex 3 contains cytochrome BC1 complex. So, the cytochrome BC1 complex, what it will do is, it will receive the electron and it will donate it to a mobile electron carrier. I will write it here. It is called as I'll just mention it here probably. Yeah, this cytochrome B6 is there, no? Sorry, cytochrome C. So this cytochrome C is called as mobile electron carrier. Why do we call it as mobile electron carrier? You see the position of this cytochrome C. It is present on the surface of the inner membrane towards the intermembrane space. So it moves from this complex to this complex, okay? From complex 3 to the complex 4. Okay, that is why cytochrome C is called as the mobile electron unit, or it is mobile electron acceptor. Remember this, which of the components are mobile electron acceptors? You should tell that it is cytochrome C. Okay, don't get confused. Now, cytochrome C donates its electron to complex 4. Now, what do we have in complex 4? We have cytochrome C oxidase. So, the cytochrome C oxidase, they also have association with copper ions. Now, what do they do is, they donate these electrons, okay, to in such a manner that they oxygen is used as the hydrogen acceptor eventually to form the water. So as the amount of hydrogen within the matrix region is removed by the water, the hydrogen ion concentration in the matrix decreases. Okay, complex 4, what is happening? They receive the electrons, they donate it for the formation of water. Now, why water? Oxygen acts as a hydrogen acceptor. It removes the excess hydrogen within the matrix, thereby reducing the proton concentration. Okay. Now, what happens? During this process, all this, there is reduction in the proton concentration in our mitochondrial matrix. But in the intermembrane space, there is very high amount of proton accumulation. Whenever there is high amount of proton accumulation, now what happens? The final complex comes, that is the complex 5. Now, the role of complex 5 is, so you can see, hold on. Yeah, the role of complex 5, what it will do is, our complex 5 has two important units. Okay, wait, let me show you somewhere. Okay. Yeah, it's all the explanation. Anyways, so what it does as, it has F0 and F1 units. Okay, what does our ATP synthetase has? It has F0 and it has F1. Okay, now what are these two important elements of our ATP synthetase? Recognizing the increase in the hydrogen concentration within the intermembrane space, now this F0, what it will do is, it acts as a channel protein to move these protons into our mitochondrial matrix. As the proteins from the intermembrane space are pumped into the mitochondrial matrix, okay, it is moved. Okay, as it is pumped, the movement of the protons, what it will do, it changes a configuration in our F1, F0. So once this configuration changes, now this will activate our ATP synthetase. As the hydrogen ions keep moving from the intermembrane space to our mitochondria, what is happening? That is increasing the ATP synthetase to convert ADP into ADP plus phosphate into ATP. So this is how the energy production takes place within our mitochondria. Now, what we have to remember is, you see, people, we have NADH, right? NADH can give us 3 ATP each molecule, and FAD can give us two ATP molecules. Okay. Now you can see how high energy rich they are. You understood, right? Can we summarize once again what is happening in electron transport chain? Electron transport chain is a series of movement of electrons by electron acceptors. Here what happens? The electron acceptors are very series, they're arranged very appropriately in the inner membrane of the mitochondria. There are 5 complexes and there are 7 electron carriers. Okay, so we have complex 1, complex 2, complex 3, complex 4, complex 5. As I've told you, these are the names of the electron carriers, important electron carriers. So how does the mechanism takes place? In the complex 1, we have NADH. Okay, it is oxidized to NAD plus. During this process, what it will give you? It will give you 2 electrons. So to move these electrons, 4 protons are pumped into our intermembrane space. Now these electrons which are received by the complex 1 is given to ubiquinone. Ubiquinol is converted to ubiquinone during this process of electron acceptance. Now we have the complex 2 where we have succinate dehydrogenase. What it will do? It will convert our FADH2 to FAD. So during this process again we get two more electrons which is given to ubiquinol. So from complex 1 and complex 2 it goes to complex 3 where it is received by cytochrome BC1 complex. So once we have this in 3, now the C3 donates the electrons to cytochrome C, which is a mobile electron transporter. So it can move from one complex to another complex. Okay. So once this cytochrome C donates it to the complex 4 releases the electron into the matrix so that oxygen can act as a hydrogen acceptor. Now you can see that how hydrogen comes in the very last place of this particular mechanism, right? So eventually it leads to the water formation. So if you see in the entire human body, we have around 80% of the water that is produced because of this mechanism. So which is fascinating, right? So again, so once we have the reduction of the hydrogen concentration in the mitochondrial matrix, an increase in the hydrogen concentration in the intermembrane space, now what happens? Because of this difference in the proton gradient, proton means positive charge. So hydrogen ions are accumulating in the intermembrane space. Now, with this increase, that will activate our complex 5, which contains F0 and F1 units of our ATP synthetase. Now, the F0 acts as a channel protein where it pumps the hydrogen ions from the intermembrane space all the way to the mitochondrial matrix. Okay. Whereas we have another unit that is the F1, which contains this enzyme called as ATP synthetase. What it does is it converts ADP into ATP, okay, by phosphorylation process, thereby creating the currencies of energy. So these are very, very important. So what I would recommend you is remember the names of the electron acceptors and how they behave. Okay. Okay. So the next topic that we did not talk properly or we did not discuss properly was understand how the anaerobic reaction takes place. All this while we understood aerobic reaction, we understood all the aerobic biochemical reactions, but we did not discuss about anaerobic reaction. So it was first studied by Shekskov. Okay, so it is also an enzymatic controlled reaction. It does not require oxygen, and the entire, if you just calculate the amount of energy output, so it's pretty less. So there are two types of anaerobic reactions based on higher organisms and lower organisms. In the lower organisms, we call it as fermentation reaction. So fermentation reaction means we have the pyruvate, which is produced by the glycolysis. Eventually, it is converted into ethanol or lactate. Okay, and then we have one more types of anaerobic reaction, which you called as lactate formation, where again you have the pyruvate that is converted into lactate. So what is happening here? If you look at the process of anaerobic reaction, we have glucose. Glucose undergoes glycolysis eventually to produce two molecules of pyruvate. Now, this pyruvate undergoes decarboxylation. Decarboxylation means what, students? A carbon dioxide molecule from each of the pyruvate is removed. Since we have two pyruvate molecules out of one glucose, so we remove two carbon dioxide. Okay. Now, what do we get? We get two acetaldehyde or acetaldehyde, two molecules of acetaldehyde or acetaldehyde. Now, by oxidizing our NADH to NAD+, what happens is acetaldehyde is converted into ethanol. Now, ethanol is used for many of the breweries like wine preparation, okay, and also many of the distilleries use the same process, okay. Since this process happens without the application or the use of oxygen, that is why we call it as anaerobic reaction. Now, where does this anaerobic reaction takes place? It takes place in the cytoplasm. Okay, remember this. Where does the anaerobic reaction takes place? It occurs in the cytoplasm. All right, students. Yes. So can we go next? Yeah. Now, if you see the entire process of our glucose formation, our yeast cannot tolerate a high about over 13% of the alcohol concentration within the medium. So there are like batch cultures which they utilize so that the yeast will not die because of alcohol poisoning due to fermentation. Okay, fine. So what about lactic acid? How does the lactic acid formation takes place? So basically, when we talk about the lactic acid formation, there is one thing that we have to remember. Basically, we can also see this in our muscle cells. Whenever we are working out, too much and we have hypoxia, like we are not supplying enough oxygen to our tissues automatically go into this anaerobic reaction. What do they do? After glucose, the glycolysis happens. After glycolysis happens, what do we get? We get pyruvate, two molecules of pyruvate, correct? So after we get two molecules of pyruvate, NADH is reduced to, NADH is oxidized to NAD plus, and during this process, pyruvate is converted into two molecules of lactate. Now, when the lactic acid accumulation happens in our tissues, that is when you start getting cramps. Okay, and that is when you have your muscles just contracted and they're not relaxing and it's an excessive pain. After some time, it will return to normal because lactate is again sent to the liver and then it's converted into glycerol. Okay, so that the lactate is getting utilized. So this is about respiration in higher plants. So let's solve some question with respect to this. If you have any doubts, students, please let me know. We can solve this. Okay. So can we go for the question rounds? So here it is. Very important questions because all of them were from previous years. Okay. So let's see. Oh, hold on. Let me share the screen. Let me share my screen. Okay. All right. So now here we have, here we have the first question. Okay, students. So let's see how much we can solve. Okay. Hold on. Yeah. So given below are statement 1 and statement 2, and we have to see which is assertion and which is reason, and we have the following options. So the first option, that is the assertion, says ATP is used in two steps in glycolysis, and the reason, the first ATP is used for converting glucose into glucose 6 phosphate, and the second ATP is used for the conversion of fructose 6 phosphate into fructose 1,6 bisphosphate. And in the light of the above statements, you have to choose the right option. Assertion is false, reason is true. Both assertion and reason are true, but the reason is the correct explanation of assertion. Okay, and assertion and reason both are false. Okay, so this is the next both assertion and reason are false, and the reason is a wrong explanation of assertion. So these are the options. Okay, so I'll give you 1 minute to answer this question. So let's see. Okay, it's very easy, isn't it? So can we solve this question? So first we have glucose, people, and we know that the formula of glucose is C6H12O6. Now, very easy, simple to remember. When the glucose was converted into glucose 6 phosphate, right? It was converted into glucose 6 phosphate. What happened here? There were 6 carbon atoms in the glucose structure. In the sixth position, this is the first carbon atom, second, third, four, okay, 5, and then we have the sixth carbon atom, students. At the sixth carbon atom, a phosphate molecule was added. If you want to add a phosphate molecule, what do? You take ATP and then you break it down into ADP plus phosphate, and then this phosphate goes into the sixth position and then it becomes glucose 6 phosphate. The same thing happens when the glucose 6 phosphate is converted into fructose 6 phosphate. So once we have fructose, okay, fructose 6 phosphate, okay, fructose 6 phosphate, you can see here fructose 6 phosphate is converted into fructose 1,6 bisphosphate. What does it mean? Already we have one phosphate at the sixth position. One more phosphate gets added at the first carbon atom. So when this happens, students, you know what it means? It means that we utilized one more ATP molecule. So this ATP is converted into again ADP plus phosphate, and this phosphate goes and attaches to the first carbon atom of the fructose. So according to our given question, when we look at the glycolysis process, we have investment phase and we have return phase, right? In the investment phase, what we will do, we'll invest 2 molecules of ATP. In the return phase, what do we get? We get 1 molecule of NADH and 2 molecules of ATP as a whole. So that is why whenever it comes to how many ATP molecules are utilized, we utilize 2 molecules of ATP in 2 steps of glucose glycolysis. Okay, so that is correct. And the reason where do we utilize is also given, that is also correct. Okay, so the correct option is both assertion and reason are true, and the reason is the correct explanation for assertion. Come on. Okay, 1 second, students. I'll just look into it. This screen is stuck one. Yeah, just a second. Sorry about it. Okay, small technical glitch. I will just, yeah, I think we are good. 1 minute. One second, students. Yeah, yes, we have it. Um, 'Yeah. So we finished the first question, right? I hope you had it right. Okay, let me see if everything is fine in the Google Meet. If you have any questions, okay, video is off. Hello. Okay, so we understood this part. So we have another question, student. Just read the questions carefully. So in this question, it's given how many times decarboxylation occurs during the TCA cycle. So thrice, many, once, twice. What is TCA cycle? Krebs cycle. Okay. How many times did we see that decarboxylation or removal of carbon dioxide taking place in the TCA cycle? Your options are twice, many, once, and then we have thrice. Okay, these are our options. Try to answer this. Yep. Can we tell the answer for this? See, students, what is decarboxylation reaction? Decarboxylation reaction states that is removal of carbon dioxide during the specific process. Okay. Now, when we talk about Krebs cycle, okay, where can we see decarboxylation? As I've told you, the Krebs cycle just now, like while we are revising, I just told you that in Krebs cycle, decarboxylation occur at two places. Where are those? One is isocitrate converted into alpha ketoglutarate, that is one place. And in the second place where it is happening, it is when alpha ketoglutarate is converted into succinyl CoA. So in these two places, carbon dioxide removal takes place. Okay, where are those? When isocitrate. Okay, I will just write it here so that you remember. Okay, just give me a second. Okay, just give me a second, students. Let me write this. See, decarboxylation. When does it happen in the first place? Once we have isocitrate converted into alpha ketoglutarate, and then we have second one, that is alpha ketoglutarate converted into succinyl CoA. CoA. So during this process, what happens? Carbon dioxide molecule is removed. So where, how does it happen? Two times. So we'll write it as two. So our option is twice. Option 4. So let's go for the next question. Fatty acids are connected with the respiratory pathway through. Okay, so we have the options acetyl CoA, alpha ketoglutric acid, dihydroxyacetone phosphate, and then we have pyruvic acid. Okay, so pyruvic acid. So this is a very interesting question. Fatty acids are connected with the respiratory pathway through acetyl CoA, alpha ketoglutric acid, dihydroxyacetone phosphate, and then we have pyruvic acid. Okay, just think about it. Did you guys get the right answer? Yes? No? Okay. Now, what happens with respect to fats? So, if we look at the fat, we will understand really how the metabolism works. So, when we look at fats, okay, I'll write it here. Sorry. Yeah. When we look at fats, okay, we have fats. And the fats, how are they metabolized? Fats are basically made up of two things. One is fatty acid and glycerol. Okay. Now, these two are broken. Now, what happens to this? This glycerol or fatty acid is eventually converted into acetyl CoA. Acetyl CoA. Now, this acetyl CoA becomes the connection link between the respiratory pathway. Correct, right? Acetyl CoA goes into the Krebs cycle, and you know what happens in the Krebs cycle, and then it comes to the electron transport chain. Okay. Whereas sometimes what happens with glycerol, the glycerol is converted into dihydroxyacetone phosphate, and again it is converted into glyceraldehyde 3 phosphate. Eventually, it has to come to this acetyl CoA itself. So, our answer for the fatty acid connecting to our respiratory pathway is through acetyl CoA. Understood, no students? Okay, fats are broken down first into fatty acid and glycerol. Eventually, the fatty acid conversion is converted into acetyl CoA, which again enters into a TCA cycle or Krebs cycle. Okay, done. What amount of the energy is released from the glucose during the lactic acid fermentation? So that's your question. Less than 7%, approximately 15%, more than 18%, and about 10%. So these are your options. So let's choose our right answer. What amount of energy is released during the glycolysis? Okay, what amount of energy is released from the glucose during the lactic acid fermentation and anaerobic reaction? Okay, so let's see. It's an easy question, right? Okay. So when we look at this question, the amount of energy released by the glucose, especially in the lactic acid fermentation, is less than 7%. That is why we say that if you look at the energy output of aerobic reaction and anaerobic reaction, it's very less in anaerobic reaction. Okay. So that is why it is a low energy yielding process or biochemical process, I can tell. Okay. So next one. What is the net gain of ATP when each molecule of glucose is converted into two molecules of pyruvic acid? Very, very important question. 8, 4, 6, 2. Okay. 8, 4, 6, 2 is your options. Okay. All right. So I would really, really appreciate you getting the right answer for this. Okay. So what happens here in glycolysis? Where do we get energy, students? Huh? Where do we find energy in glycolysis? At what points? One is 1,3 biphosphoglycerate is converted into 3-phosphoglycerate. That is where 1 ATP is produced, right? And other one is produced when we have phosphoenol pyruvate converted to pyruvate, two places. But when you look at the question itself is a little bit tricky. Why the question is a little bit tricky is because I will tell you. They are asking about the net gain. Okay, where are we? Yeah. What is the net gain, not the gross gain? What's the difference between net gain and the gross gain? See, investment. When I invest something, I know that while converting glucose into glucose 6 phosphate, I use 1 ATP. Again, fructose 6 phosphate, right? Into fructose 1,6 bisphosphate. I get how many? I use one more ATP. So I have used around 2 ATP, 2 ATP used. Whereas, as I told you, when I just converted in 3 phosphoglycerate into phosphoglycerate or phosphoenol pyruvate into pyruvate, each 3 phosphoglycerate, what I told 3 phosphoglycerate into, or wait, when I convert, for example, phosphoenol pyruvate, right? So when I look at phosphoenol pyruvate, I have 2 molecules of phosphoenol pyruvate. Each molecule of phosphoenol pyruvate becoming into pyruvate is giving me how many? 2 ATP. Like that, there is one more thing, one more place that is happening. Where is that? Another place that is happening. I just mentioned you. What did I tell you? That if you look at one more place, that is 1,3 biphosphoglycerate, when it is converted into 3 phosphoglycerate, there also we get 2 ATP molecules for one molecule. Why? Because you know that when the glucose is broken down, it gives rise to 2 halves, 3 carbon containing, 3 carbon containing. That is why we have 4 ATP as the gain here. 2 plus 2 is 4 ATP, right? So how much are we investing? 2. 4 minus 2, 2. So our answer is 2. Okay. We get only 2 ATP molecules out of the glycolysis. Next one. Identify the cytochrome which acts as a mobile carrier for the transfer of electrons between complex 3 and complex 4. Your options are cytochrome A, cytochrome A3, cytochrome B1C, cytochrome BC1 complex, or cytochrome C. So these are your options. So this is a very, very easy question. It should be an easy question because we just discussed not too long ago. It was just a while ago. Okay. Okay. I told you to remember one important thing. Even though you cannot remember the concept, but sometimes you have to memorize some things. Whenever mobile electron carriers come, you directly go for cytochrome C. What does cytochrome C? We know that we have complex 1, complex 2, complex 3, we have complex 4, and then we have complex 5 in electron transport chain. Now look here, when the electrons from complex 3 to complex 4 has to go, we have something called as site C or cytochrome C, which is present on the surface of the inner membrane. Now what does this site C? I told you they are mobile electron acceptors. They can move from cytochromes, I mean like complex 3 to complex 4. The mobile means what? Movable. So when they move, what do we get? We get cytochrome C. Understood? So answer is cytochrome C. And cytochrome B1C is a complex for the cytochrome. It's a complex for third complex. Cytochrome A3 is for 4. Okay, and cytochrome A is also for fourth complex. So that is why cytochrome C is the correct answer. The number of times the decarboxylation of, I think we just answered this. Okay, next one. What is the 5 carbon compound formed during the TCA cycle? Okay, alpha ketoglutric acid, then we have oxaloacetic acid, then we have succinic acid, and then we have fumaric acid. Okay, so tell me which is your right option. Which is the 5 carbon containing compound that is formed during the TCA cycle? Okay, maybe you don't know, but the answer for this is alpha ketoglutric acid. So alpha ketoglutric acid is the only 5 carbon containing compound that is formed during the entire TCA cycle or Krebs cycle. This also you just memorize. Rarely these type of questions are hard to come, but when its structures are there, you have to just like take a look into it. Okay, chalo. So let's solve another question. Which of the following statements is incorrect? ATP is synthesized through complex 5. Oxidation reduction reactions produce a proton gradient in respiration. During aerobic respiration, the role of oxygen is limited to the terminal stage. The electron transport chain, one molecule of NADH plus H plus give rise to 2 ATP molecules and one FADH to give rise to 3 ATP molecules. So we just have to choose which is the incorrect option. So just go through the options carefully now and then see which is your right option. Okay. Just read the question, students. Yeah. So did you go through all the options? So can we see what's the right option here? See, ATP is synthesized through complex 5. Yes, complex 5 has F0 and F1 units, which has ATP synthetase and it will produces ATP. Correct. Oxidation and reduction reaction produces the proton gradient. Yes, oxidation is happening. That is why the electrons are produced and it is reducing. That is why the hydrogen accumulation is happening. Okay, so that is why it's called as oxidation and reduction process. That is the electron transport chain. That is right. Okay. Okay. Sorry. Yeah. Next one. Sorry. Next one. During the aerobic reaction, the role of oxygen is limited to the terminal stage. Exactly. At the complex flow, what happens? Oxygen acts as hydrogen acceptor. So they finally give rise to what? Water. So that oxygen just removes the more amount of hydrogen that is present in the matrix. Apart from that, it does not have greater role, but you know how much it is important physiologically, right? Next one. In ETC chain, one molecule of NADH plus H plus give rise to two molecules of ATP. This is wrong, right? NADH gives rise to how many molecules of ATP? 3 ATP. And then what about FADH2? FADH gives rise to 3 ATP they have given. It is actually 2 ATP. Okay, you just have to reverse this. So that's a small confusion. We just forget this while we are doing it in a hurry. That's why we have to concentrate a little bit. Okay, next one. The number of substrate level phosphorylation in one turn of citric acid cycle. How many substrate level of phosphorylation is happening in the citric acid cycle? 1, 2, 3, and 4. What is your option, students? Substrate level phosphorylation. I told you what is phosphorylation. Adding phosphate, right? Where is it happening? How many times? Do we have an answer here? Okay. So if you look at the entire TCS cycle, where do you find ADP is converted to ATP or GDP is converted to GTP? How many times you come across? One time. So the substrate level of phosphorylation, if you come across, it's only one time. Okay. So that is why your option should be one. You just see like where all the ATP is coming out or where it is going in. So that is when we understand the process of phosphorylation. So it happens once in the citric acid cycle. So we take it one. Pyruvate dehydrogenase activity during the aerobic reaction requires calcium, iron, magnesium, cobalt. Okay. So which is your answer, students? So for the pyruvate dehydrogenase to perform during the aerobic respiration, okay, what of these ions are required? Calcium ion, iron ion, cobalt, or magnesium ion? Okay. So tell me which ion. Very, very important question, you know. I know it's such an easy question because when we discuss about this, the way the pyruvate dehydrogenase work, we really mention it in the equation, correct? So we did mention in the equation. Now tell me, where does it take place? Like what does it require? Yes, your answer is correct. So the students are saying the answer is magnesium. Yes, magnesium is very, very, very important. Okay, because it helps in the activity of pyruvate dehydrogenase. It's just like a what do you tell? Cofactor. It's a cofactor for the pyruvate dehydrogenase to function. Okay, so magnesium ion. Next one, conversion of glucose into glucose 6 phosphate. The first reversible reaction of glycolysis is catalyzed by the entire glycolysis process. You have one enzyme that is mentioned, one of the important enzymes that is mentioned in your textbook, and it is there. So let's see how many of you will get the right answer. Phosphofructokinase, aldolase, hexokinase, enolase. During the process of conversion of glucose 6 phosphate into glucose into glucose 6 phosphate, which is an irreversible reaction, which enzyme catalyzes this reaction? So that's our question. Do we have an answer? Yes, perfect. Yes, students, you are absolutely right. Your answer, that is hexokinase, is perfect, 100% correct. Okay, so hexokinase is the one that helps in conversion of glucose into glucose 6-phosphate. That's the only enzyme that is given, so you go for that. Okay, another question. Respiratory quotient, RQ value of tripalmitin is. What is respiratory quotient? Amount of carbon dioxide used to the amount of carbon dioxide released divided by amount of oxygen used for any substrate. We have proteins, carbohydrates, and then we have proteins, carbohydrates, and fats as the substrates. So when we want to burn any of this, we try to take the respiratory quotient, correct, right? So how much of the tripalmitin gives? What is its value? 0.09, 0.9, 0.7, 0.007. So what is it, people? Okay. See, when we talk about respiratory quotient, I told you amount of carbon dioxide released divided by amount of oxygen consumed. Sorry, amount of oxygen consumed. Now, if I take 1 second, I'll just erase this amount of oxygen consumed. So now, see students, whenever I take 3 examples, one is carbs, next one is fats, other one is proteins. Okay, this is very important value and very easy to remember. When you take carbohydrate like glucose, when you burn them, the amount of oxygen taken and the amount of carbon dioxide released is equal. So when we divide anything, 2 by 2 is what? 1. Similar to that, in carbohydrates also you'll get 1. But in fats, the respiratory quotient is little less, so it comes around 0.7. In proteins, it is little less than the carbohydrates but higher than the fats. It comes around 0.9. Very important, very easy. So remember these values for the fat. Tripalmitin is a fat, so it's 0.7, students. Okay, so do we have one more? Yeah. Which of the following statements regarding the mitochondria is incorrect? Mitochondrial matrix contains single circular DNA molecule and ribosomes. Outer membrane is permeable to monomers of carbohydrates, fats, and proteins. Enzymes of electron transport are embedded in the outer membrane. Inner membrane, okay, is convoluted. 1 second, I can't read this. Okay, yeah, convoluted with in infoldings. So which of the following regarding mitochondria is incorrect, students? Just look at it. What do you think? It's a very simple question, isn't it? It should be. Yeah. So let's look at the first option. Mitochondrial matrix consists single stranded DNA molecule and ribosomes. Yes. Outer membrane is permeable to monomers of carbohydrates, fats, and proteins, as I told you. This is also correct. Enzymes of ETC are embedded in the outer membrane. Is it correct, people? No, this is wrong. So this is actually inner membrane, correct, right? All right, coming to the fourth option, that is inner membrane is convoluted with infolding. Yeah, that's what causes cristae formation, and that's why we have this option is correct. So our right option for this question is option 3. So okay, so this is it, students. We will wind up, and we have come to the end of our PowerPoint. Okay, chalo. So thank you so much, students. I'll come in the next class and we'll discuss about the next chapter. All the best for your pre-boards. Do well. I'll see you in the next class. Thank you.